Welcome to the Chapter 8 video. Today, we discover the world of cellular reproduction. So, here we go. There are different kinds of cellular reproduction. There's asexual reproduction, where there's no sex needed. And this is where cells produce genetically identical cells. And this is done through a process called mitosis. And then there's sexual reproduction. And this requires two different sex cells, a sperm and an egg. And the process generally used to produce a sperm or an egg is called meiosis. A sperm or an egg has different genes from each other and the parent. They are not identical. So the cell that produces the sperm or the egg is not identical to the sperm or the egg. And the bonus of having a sperm or an egg being genetically different from the parent cell is that it sort of becomes a mix of genes. And so when you have two people have sex and make an offspring, the offspring is sort of a shuffling of their genes in order to provide new combinations of genes. It helps increase the variation in the population. We'll talk about why that's healthy towards the end of this lecture. But meiosis, as I said, is the process in which you make sex cells, sperm and an egg. And sex cells are all about sexual reproduction. So, first thing we're going to do is talk about asexual cellular reproduction. And this is cell division, mitosis. Cell division makes new cells. It duplicates a cell, specifically. This means that mitosis creates two identical, genetically identical cells. And we call them sister cells. You'll find throughout this chapter that the scientists who first discovered these processes had a mild obsession for the female gender. And so everything is mothers and daughters and sisters, which is weird, but okay if they're into that. Anyway, the cells produced through mitosis have the exact same DNA and the exact same cytoplasmic organelles. So let's talk about some important physiological features of cells so that we can more accurately talk about mitosis. We can use the correct vocabulary. So, the genome is the sum of DNA inside the cell. All of the DNA in the cell is the genome. DNA is long nucleotide chains. Remember your adenine, your guanine, your cytosine, your thymine. Those are all nitrogenous bases, and a single nucleotide has a single nitrogenous base. DNA is long chains of A, G, C's, and T's, of nucleotides. And DNA carries genes. Genes, as we'll talk about more in chapter 10, code for proteins. So we find DNA in two different conditions. Either uncondensed, which I'm pretty sure isn't a word, but I'm making it. So uncondensed or condensed. In other words, you can find DNA in sort of a loose, unraveled state or a highly compact, condensed state. Chromatin is the term for that loose, unraveled, uncondensed state. So we have long DNA strands sort of looping around in the nucleus. Looks kind of like a ball of yarn after a cat's played with it. So it's uncondensed. And it's made up of the DNA and protein packaging. So the protein packaging is what's creatively known as the histones and the non-histone proteins. So these are the proteins inside the nucleus. So the chromatin that you see is those long chains of DNA wrapped around histones and non-histone proteins, creating sort of these loose, ribbony packages. We can contrast chromatin with the highly compact organized, condensed chromosomes. So this is when chromatin is wound and coiled up, compacted together, condensed into chromatids. So a chromatid is a rod-shaped structure, highly compact. All the DNA in a particular strand has been compacted, wound up, tightly organized into a rod shape. It's condensed. Humans have 44 chromosomes and then 2 sex chromosomes. You might be familiar with the sex chromosomes, the X and the Y, right? A female has 2 X chromosomes and a male has an X and a Y. So any given human has 44 chromosomes and then an XX or an XY, depending on their gender. So here we go. This is just straight DNA. And then for chromatin, we wrap it around some histones like this, and we sort of wrap it up into these loose strands here. So this single chromatin loop here is 100,000 base pairs. And when you highly condense our chromatin into chromosomes, you get this right down here, this structure, which is this X shape. This is 2 rods. So this is one of those chromatid rods I was talking about. And you can see this loop here, which is 100,000 base pairs of DNA, is this tiny little section right here in the chromatid. So a chromatid, a single one of these 2 rods, a chromatid, is very highly organized, highly compact. Okay, important vocab. Diploid. Diploid and haploid are words we're going to be using, and they refer to the number of chromosomes carried by an organism. Diploid means you have 2 sets of chromosomes, or rather, for each set of genes you have 2 chromosomes. One came from your mom, one came from your dad. So your chromosomes exist in pairs. Probably the best way to say it there. Your chromosomes exist in pairs. So one from each parent. And we can contrast that with haploid, where we have a single set of chromosomes. So they are not pairs. Haploid cells are only ever seen in humans and in other animals in the sex cells. So a sperm or an egg is haploid. Every other cell in an organism is diploid. This isn't true for every organism. For instance, when you look at a moss, chances are what you're looking at when you see that moss is haploid. But for animals, at least, every cell except the sperm and the egg is diploid. Sperm and the egg are haploid. So for humans, we have 44 chromosomes plus the sex chromosomes for a total of 46 chromosomes. So our diploid number of chromosomes is 46. Our haploid number of chromosomes is 23. Human cells can also be described as somatic, which are the non-sex cells. So anything that isn't a sperm or an egg is a somatic cell. And all somatic cells are diploid. So like I said, 46 chromosomes, 44 plus an XY or an XX, depending on your gender. XY male, XX female. And somatic cells are produced through mitosis. So if it's a somatic cell, it was generated through mitosis. We can contrast somatic cells with gametes. Gamete is the term for the sperm or the egg, the sex cell. So sex cells are always haploid. So humans have 46 chromosomes. Their sex cells have 23 chromosomes, 22 non-sex chromosomes, and then a single X or a single Y, because they have half the number of chromosomes. Gametes are always produced through meiosis. So, starting off with mitosis, let's hit up some of that vocab. Mitosis is cell division. Cell replication, cell multiplication, cell division. Same thing, mitosis. That's right. Biology is the only science where multiplication and division mean the same thing. Ha 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 ha. I'm so hilarious. Shut up. Sister chromatids. So, the official chromosome, as we saw in that earlier picture, is two of those rod-shaped chromatids put together. It's a pair of duplicated chromatids. Being chromatids, this is condensed DNA. So, it's important to note when I say duplicated, I mean genetically identical. So, sister chromatids are two identical chromatids joined together at the centromere. Oh, look at that. The centromere is made of DNA, and it joins the sister chromatids together to make a vague X shape. So, two sister chromatids joined together at the centromere make a chromosome. The aster. The aster, if you remember way back to the chapter on the organelles you find in a cell, when we talked about the cytoskeleton, the aster is the structure where microfibers and microtubules are constructed. So, aster plays a big role in mitosis. So, this is star-shaped microtubules around a centriole pair. The centrioles are sort of like a little ring, and there's two little ring structures surrounded by microtubules. And so it sort of looks like two little ring structures with a starburst of microtubules coming out around them. And in fact, aster is Latin for star. So, amazingly enough. The kinetochore, besides being one of those awesome science words that you would never use outside of talking about mitosis, it is proteins on opposite sides of the centromere. I have pictures for you to make this all make far more sense. So we'll get to those in a moment. But the kinetochore are little proteins on either side of the centromere. So if you think of your chromosome, right, we've got identical sister chromatids, and they're held together in this X shape by a centromere, which is my thumb and forefinger all of a sudden. I'd have little proteins sticking off here and here on either side of the centromere. Bam, that easy. Then we have the mitotic spindle. So the mitotic spindle is important. This is made of microtubules. The microtubules of the mitotic spindle are produced at the aster. Remember how I said the aster has a very important role in mitosis? Well, the asters produce the mitotic spindle. And the mitotic spindle is just long microtubules that attach between the aster and the kinetochores. Finally, we have chromatin, which is unraveled, loose, uncondensed DNA in the nucleus. So here we go. This is a single chromatid right here. This is two identical chromatids, or two sister chromatids, joined together by the centromere, which is also made of DNA. Here's another view of two sister chromatids joined together by the centromere, which is this circle here, and they've helpfully put the kinetochores coming off of this sucker. And then over here, we have an aster, and as you can see, this is those two sort of ring-type structures here. They're more represented as cylinders in this image, but whatever, we'll go with it. And they're surrounded by a starburst of microtubules, hence the name aster, Latin for star. And anyway, you have two asters, and each aster is cranking out microtubules, and these microtubules are attaching to these kinetochores. These microtubules are the mitotic spindle. So the cell cycle lasts from when the cell is first formed to when it finishes dividing. And there are important phases of the cell cycle to discuss. Interphase is where a cell spends 90% of its life cycle. If you see a cell, there is a 90% chance that what you are looking at is in interphase. Interphase consists of three separate stages. G1, in which the cell functions and grows normally. When we talk about how a brain cell sends signals, it sends its signals during G1. When the glands that produce sweat, the cells that are making up that gland are in G1. G1 is when a cell is doing its thing, when a cell is functioning. Cells in G1 have the diploid number of chromosomes. So, the diploid number of chromosomes. And that DNA is present in the form of chromatin, loose and uncoiled in that nucleus. G1 also has the purpose of collecting enough nutrients and energy for the cell to enter into the next part of interphase, which is the S stage. So the S stage or S phase of interphase is named S for synthesis. What is it synthesizing? What is it making, you ask? That's right, DNA. So the chromatin is duplicated during the S phase. All of the DNA in the nucleus is copied. So it is actually in the S phase, by the end of the S phase, when we have the nucleus filled with double the normal amount of DNA. Two exactly identical copies of the genome floating around, crowded together in that nucleus. Again, it's still in the form of chromatin. In S phase, we also have the duplication of the centriole pair. So the centriole pair, with the little microtubules coming out from around it, is an aster. So duplication of the centriole pair is duplication of the asters. So in other words, we make a second aster. So in the S phase, we synthesize an entire copy of our DNA, and we synthesize another aster. And this leads us to G2. G2 is the third and final stage of interphase. In G2, the cell gathers some more nutrients in preparation for the M phase of the cell cycle, or the mitotic phase. 10% of a cell's life is represented by the mitotic phase. The mitotic phase is when the chromosomes condense, so the chromatin condenses into chromosomes, and cell division occurs. Here we go. The green, blue, and yellow, G1S and G2, all represent interphase. You can see it's the grand majority of time in the cell cycle. Then we have this little red section here that's the M phase. So the cell status in G2, which is important because I might just have a question on an exam where you have to, be able to draw all the stages of mitosis and draw what a cell looks like in G2. So a nuclear envelope is present in a cell in G2. The DNA is in the form of chromatin. There's double the amount of DNA in there, and it's all unraveled and loose, and because of that, we see the nucleolus. Remember, the nucleolus is a dark spot in the nucleus, and that dark spot is not a structure, more of a concentration of DNA. So if you think of a ball of yarn after a cat plays with it, chances are the cat won't have destroyed the entire ball of yarn. So if you gather it all up into a pile, you'll have a bunch of loose, sort of unravelly, uncoiled yarn, and then what's left of the ball in one spot, a nice concentrated spot. So that's the nucleolus, a concentrated spot of chromatin. Not a structure, not an organelle. In G2, we have count them, two asters. We also have, like I said earlier, double our DNA. We have duplicated our chromatin. It's uncondensed and it's identical. And now this cell is ready for mitosis. So here's a picture of a cell in G2. We have an intact nuclear envelope. We have all this chromatin here. I have a nucleolus over it here. And we have two asters. So, bam. Know that. All right. Now it's time for mitosis, or rather, mitosis in the next video. See you there, kids.